Wonderful. Okay. Okay, welcome everybody. Thank you all for coming to our first Associates Flash Talk session. My name is Kelsey Miller. I'm a postdoctoral research fellow at the Massachusetts Institute of Technology, and I will be moderating the session along with my colleague, Dr. Deborah Brada Das, a postdoctoral associate at UTMD Anderson Cancer Center. We're very happy to be here today and to highlight the amazing research of our associates. Each associate's pre-recorded presentation will be about 5 minutes, and after watching all of the associates' flash talks, there will be about 30 minutes of Q&A. At any time during their pre-recorded talks, please post your questions in the Q&A box on your screen. Since time is limited, it is likely we won't be able to get to everyone, so please try to keep your questions succinct. The moderators, me and Deba, will paraphrase and or combine your questions if we observe a theme. And without further ado, let's give our attention to our associates. They are Krista Friedman, Jared Mayers, Nicole Perry-Hauser, Zhuangteng Liu, Swati Gupta, Lydia Germay, Yuxuan Mao, Ahmad Naban, Joseph Swift, and Coral Zhao. Thank you all and enjoy.
You went to the hospital, where the doctor took a sample of the infection, tested it for antibiotic susceptibility, and sent you home with a prescription that eliminated the infection. Now imagine that after your full course of antibiotics, instead of a full recovery, you feel worse than ever. You repeat the process, trying a stronger dose of a different first-line antibiotic, but it doesn't work. Running out of options, you're prescribed a second-line antibiotic that not only doesn't control your infection, but also leaves you with permanent hearing loss. You're now one of the millions of Americans in a life or death battle against a drug-resistant microbe. It's estimated that by 2050, over 10 million people globally will die each year from a drug-resistant bacterial infection. Despite this sobering fact, the rate of development of novel antibiotics is at rock bottom. To tackle this problem, scientists, including those in the Hatfill lab where I'm a postdoc, are turning to the bacteriophage. Phages are viruses that infect and kill bacteria. Discovered over 100 years ago, phages are making a comeback as we explore their potential to serve as highly specific, self-replicating, shelf-stable, and cheap-to-make alternatives to conventional antibiotics. In 2019, our lab teamed up with physicians to treat a pediatric cystic fibrosis patient battling a drug-resistant infection of Mycobacterium abscessus. This successful treatment was enabled by our collection of thousands of mycobacteriophages, phages that specifically infect mycobacterial hosts. This collection is the product of C-Phages, a global research experience for first-year undergraduates founded by my advisor, Graham Hatfill. Among the C-Phages collection, our team found 3 phages able to efficiently infect and kill the bacterial strain colonizing the patient. Within weeks of well-tolerated IV infusions, this patient began to show signs of a remarkable recovery, and within months was able to return to school, get a puppy, and learn to drive. Since then, dozens more patients have benefited from compassionate use mycobacteriophage therapy and gone on to resume the activities that give them joy. However, treatment is not always successful. One limitation can be the patient's development of antibodies against the phage. These antibodies bind to the phage soon after it enters the bloodstream, contributing to rapid clearance by immune cells. Those phages that do circulate long enough to reach the targeted pathogen face another challenge. The antibodies can bind to the tail tip and tail fiber proteins that are essential in the interaction with the host bacterium. These antibodies therefore neutralize the phage and prevent it from infecting. One potential way to recover from such an occurrence is to engineer the phage to remove or mutate the parts that antibodies bind to. These immune-evasive mutant phages would have restored therapeutic potential. To advance this idea, I've been generating atomic resolution structural maps of bacteriophages. These experimental structures let me identify highly immunogenic phage components, which I can then engineer. Take, for example, this phage, BXB1. Using a Western blot, I showed that the capsid and tail tube proteins are immunodominant. That is, all the antibodies raised against the phage in mice bind to the capsid and tail. By solving BXB1's structure with cryo-EM, I discovered that these surfaces are not smooth. Instead, both the capsid and tail are decorated by protruding protein domains, shown in purple. I used genomic engineering to remove these domains from the capsid and/or tail proteins, creating mutant phages Phi-FW1 and FW2. These mutations render the mutant phages invisible to the antibodies that bind BXB1. Utilizing these strategies, I aim to structurally engineer made-to-order phages with reduced immunogenicity, expanded host range, improved stability, and beyond. With that, I thank you for your attention and gratefully acknowledge the support of my mentoring team, Graham Haffel, John Williams, and James Conway. I also thank the CPhages program for enabling this science, and my lab mates and collaborators for making it fun. Finally, I thank my funding sources, especially the National Institute of Allergy and Infectious Diseases, which awarded me a K-99 to further develop structure-guided immunoengineering of phages. Think back to the last time you had a bacterial infection. Maybe it started as just a cough, but
Hi, my name is Jared Mayers, and I'm a postdoctoral fellow in the laboratory of Emily Balskus at Harvard University. I'm excited to tell you today about my project entitled The Identification and Targeting of Microbial Putrescine Acetylation in Bloodstream Infections. Antimicrobial resistant, or AMR, bacterial infections represent an escalating global public health crisis predicted to cause 10 million deaths by 2050. Bloodstream infections, or BSI, are a primary driver of this trend. Treatment options for BSI and AMR infections more broadly are limited by both a narrow repertoire of targets and challenges in drug development that have slowed the pipeline of new antimicrobial agents. Exploiting disease-associated metabolism for clinical benefit has proven promising in contexts such as cancer and Mendelian disorders, but this paradigm is underutilized in bacterial infections. We hypothesized that by identifying bacterial metabolic behavior specifically in the in vivo context, we could highlight previously underappreciated activities that play causal roles in disease and are thus more likely to serve as potential therapeutic pressure points. To test this hypothesis, we performed targeted metabolomics on plasma samples from patients with gram-negative BSIs and controls. This revealed significant differences in 62 metabolites between the two groups, 8 of which were associated with increased mortality, highlighted here in black and blue. Unexpectedly, 2 of these 8 metabolites, N-acetylputrescine and 4-acetaminobutanoate, highlighted in blue, were from a specific metabolic pathway, putrescine metabolism. To determine whether these putrescine metabolites were bacterially derived, we turned to a mouse model of BSI, which involves injection of a slurry of mouse stool from the cecum. For this experiment, we compared plasma metabolomics profiles of mice receiving an injection of PBS, a heat-killed cecal slurry, which is a sterile injury, and cecal slurry with live bacteria. We hypothesized that levels of microbial products would change only in the presence of live bacteria. And indeed, N-acetylputrescine was elevated only in those mice receiving live bacteria. Across kingdoms, putrescine and related polyamines play diverse roles in cellular homeostasis, and high levels of these polycationic molecules can be toxic. So their levels are carefully regulated at the level of synthesis, import-export, and charge neutralization, although the enzyme performing this latter function in the case of putrescine was unknown. SPEG quickly emerged as the likely candidate responsible. It belongs to a class of structurally diverse enzymes whose activity towards longer chain polyamines such as spermidine have been biochemically validated across kingdoms. Unfortunately, prior work had explicitly excluded putrescine acetylation activity, possibly because physiologic substrate concentrations were not studied. By examining an E. coli SPEG mutant previously generated as part of an international knockout collection, we confirmed that SPEG deletion resulted in a loss of N-acetylputrescine production, which was rescued by genetic complementation. Kinetic characterization of SPEG demonstrated activity towards putrescine at the high end of physiologic intracellular levels in E. coli. This was above concentrations previously assayed, indicating how this activity could have been undetected. Having identified the enzyme responsible, we next wanted to ask whether this metabolic activity was important. Using an inducible CRISPR-I interference-based system, we confirmed that SPEG loss significantly impaired proliferation in culture. These findings also extended to our in vivo cecal slurry model a bloodstream infection, where we found that repression of SPEG resulted in a significant delay in mortality compared with controls. Combined with our human and mouse data, these findings support the hypothesis that microbial metabolic products detected in infection can highlight pathways that play important roles in pathogen physiology. We next sought a chemical method of inhibiting bacterial putrescine acetylation. We figured out that diminazine, a veterinary antiparasitic agent, blocked SPEG activity. Then, through a series of experiments I unfortunately don't have time to go into, we determined that inhibiting SPEG increased the permeability or leakiness of bacterial membranes. And by way of explanation, gram-negative bacteria have a double plasma membrane on either side of their cell walls, which is why you see measurements of both membranes here. Now, this is cool because two primary mechanisms of antibiotic resistance are limiting antibiotic entry and increasing efflux. So if inhibiting SPEG leads to membranes that are leaky, perhaps we could overwhelm these defenses. Indeed, we can. This is with a tetracycline-resistant clinical isolate of E. coli. And you can see that SPEG inhibition results in increased intracellular accumulation of the drug. Even more exciting, this observation extended in vivo, where combination therapy increased the median and overall survival of mice with the same tetracycline-resistant E. coli in our cecal slurry bloodstream infection model. So as I mentioned up front, given the rising challenge of confronting AMR infections, new and creative strategies are needed to replenish our dwindling pipeline of antibiotics. Guided by our observation of N-acetylputrescine elevations in BSI, we identified polyamine acetylation by SPEG as a prominent microbial metabolic activity during infection and demonstrate that blocking this activity impairs pathogen fitness. This susceptibility occurred at least in part by increasing membrane permeability, which improves sensitivity to existing antibiotics. This strategy directly circumvents multiple modes of resistance utilized by pathogens to limit intracellular access of drugs and potentially offers an exciting way to extend the lifespan of current therapies. And with that, I have many people to thank, most notably my mentor, Dr. Emily Balskis, and the team of folks in her lab that I work with, as well as my collaborators and funding sources. Thank you.
Hi, my name is Nicole Perry-Hauser, and I'm a postdoctoral research fellow at Columbia University. My long-term research interests involve resolving signaling pathways downstream of adhesion GPCRs and establishing how or if these receptors' adhesive properties influence signaling events, and in turn, whether signaling impacts synapse formation and neuronal wiring. For today's Flash Talk, I would like to share work from a recent publication that investigated the repulsive role of latrophilin-2 in the mouse hippocampus. Adhesion G-protein coupled receptors are the second largest family of GPCRs. There are 9 subfamilies, and my work to date has focused on subfamily 1, the latrophilin receptors. ADGRLs are highly expressed in the nervous system. They're associated with neuropsychiatric diseases, notably ADHD and substance use disorder. For the work presented today, I want to highlight the role of these receptors in promoting synapse formation and neuronal circuit assembly. During my postdoc, I worked to develop a toolkit of ADGRL constructs that allow me to isolate different structural and functional features of the receptor, with the intention of using them for eventual in vivo analysis. Here, I'm showing the adhesive domains for the latrophilins, the N-terminal lectin domain, or RBL, and the ULF domain, which are responsible for mediating cell-cell interaction. These receptors also contain a conserved GPCR autoproteolysis inducing domain, or GAIN domain, which is essential for autoproteolytic cleavage, which occurs at the GPCR proteolysis site, dividing this receptor into 2 fragments, an N-terminal and C-terminal fragment. These remain associated via non-covalent interactions throughout receptor biosynthesis and trafficking to the plasma membrane. This receptor cleavage generates a peptide stretch directly following the proteolytic cleavage site, known as the Stockel, or tethered agonist. Exposure of the tethered agonist results in adhesion GPCR activation and downstream signaling through peptide interaction with the 7-transmembrane domain of the receptor. My recent publication with collaborator Dr. Lixin Luo at Stanford University demonstrated the feasibility of using these constructs that isolate specific structural and functional elements of ADGRL in vivo. Briefly, we took advantage of an assay established in the Luo lab that showed repulsive interaction between endogenous ligand, tenurin-3, and ADGRL2 is required for precise hippocampal assembly. Notably, reciprocal repulsions mediated by the tenurin-3 ADGRL2 interaction instructed proximal CA1 axons to target the distal subiculum, and more distal CA1 axons to target the proximal subiculum. Here, I'm showing these topographic connections. Tenurin 3 positive proximal CA1 axons are repelled from latrophilin 2 expressing proximal subiculum, and latrophilin 2 positive axons are repelled from tenurin 3 expressing distal subiculum. We used this model to develop a misexpression assay. Here we express lentivirus of Cre-ADGRL2 in the proximal CA1 at postnatal day 0. This was followed by injection at P42 of a Cre-dependent membrane-bound M-cherry into proximal CA1, which functioned as an axon tracer. Misexpression of ADGRL2 in the proximal CA1 neurons caused mistargeting of the axons to the proximal subiculum. Here you can see the fraction of total axon intensity within the proximal, medial, and distal subiculum. With such robust differences, we could use this assay as a platform to interrogate whether functional mutants of ADGRL2 are required for axon targeting. We first expressed an ADGRL2 mutant with a disrupted tethered agonist, and when we did so, we failed to misdirect the proximal CA1 axons to the proximal subiculum. As depicted here, you can see that the axon intensity increases, similar to our negative control Cre-expressing virus. However, when we misexpressed an ADGRL2 mutant with disrupted autoproteolytic cleavage, we observed a phenotype similar to the wild-type receptor, again depicted here. Thus, we showed that tethered agonist activity, but not autoproteolytic cleavage, was required for ADGRL2-mediated axon repulsion. This validated the use of receptor construct with isolated structural and functional elements in vivo. In an independent position, I would like to use a similar approach to study how specific structural and functional elements of ADGRL3 impact synapse formation in the striatum. Thank you for your time. I'd like to thank my mentor, Dr. Jonathan Javich, as well as the main collaborators on this project, Dr. Lichin Luo and Dr. Daniel Pedrick.
The brain has a structure that is far more complicated than any other tissues. I'm Xiaotang Lu, a postdoc at Harvard working in connectomics. This is a new field of neuroscience that attempts to reveal the brain's complexity by describing all the neurons and their synaptic connections using serial electron microscopy. To map a connectome, we begin by staining brain tissues with heavy metals. After staining, the tissue is embedded in resin. We then cut serial ultrathin sections and image them using EM. Once imaged, the sections are stitched and aligned. We then segment cells from the image volume. While this was done manually in the past, we now often use machine learning to accelerate this process. Segmentation eventually leads to 3 reconstructions of neurons. Thanks to the high resolution of EM and the ubiquitous heavy metal staining, we can see all cellular and subcellular details, including neuron-to-neuron connections via synapses. Here you can see a green neuron is making a synapse with a red neuron. Synapses can be easily identified by vesicles in the membrane thickening on the EM image. However, we are facing a challenge with EM imaging. Neurons are not generic. In fact, they display remarkable molecular diversity, which underpins their unique ways in processing and transmitting information. So a critical question arises. How do we differentiate the neuronal types in these black and white EM images? Can you tell which neurons are inhibitory, which are excitatory, and which are non-neuronal? Immunostaining with fluorescent antibody is a powerful method to identify cells based on their biomolecular signatures. However, antibodies, due to their large size, cannot permeate intact cell membranes. We often use detergent to partially dissolve these membranes to allow antibodies to access intracellular targets. This process, although effective, compromises the tissue's ultrastructure. As shown here, a detergent-treated tissue can be so altered that it's impossible to segment cells. During my postdoc, I discovered that small immunoprobes, like nanobodies derived from heavy chain-only antibodies, can penetrate cells without the need for permeabilization. You can see here anti-GIP nanobodies accurately bind to YIP, a mutant GIP, without the use of detergent. And because of that, the tissue ultrastructure was well preserved after nanobody staining. This discovery paved the way for native a method that allows immunofluorescence and EM image on the same tissue. It enables superimposing the fluorescent immunolabels directly onto the EM images to aid cell type identification. In this image, you can differentiate astrocytes, microglia, and endothelial cells, and they were marked by different colors. We recently discovered that single-chain variable fragments from monoclonal antibodies function similarly to nanobodies in facilitating detergent-free immunostaining. And this allows us to leverage sequence antibody libraries, including those from our collaborator from Neuromap at UC Davis. They've engineered many neuronal antibodies into single-chain Fs and conjugate them with different fluorophores, thus achieving a 6-color immunofluorescence imaging of a mouse cerebellum and correlating with EM. However, as transcriptomics continue to discover new biomarkers related to cell diversity, we face a significant bottleneck in producing small immunoprobes to identify them. My recent work suggests aptamers might be the solution. The DNA sequences, when folded into tertiary structure, could bind to target proteins with remarkable specificity. Aptamers also allow for efficient in vitro selections of novel biomarkers from vast synthetic libraries of randomized DNA sequences. As shown here, we've successfully used aptamers to identify canonical markers for astrocytes and oligodendrocytes, as well as newer markers for them. They are also effective in labeling various neuronal types. All these images were obtained without the use of detergent. And I believe the tools I described today will help foster a more comprehensive understanding of the brain's molecular and circuit architecture. With that, I want to thank all my collaborators and my funding from Brain Initiative K99 Award and the symposium for providing me such a great opportunity to present this work. And with that, I would like to take any questions.
Hello, everyone. I'm Dr. Swati Gupta, a postdoc in Benson and Huntley Lab, and I want to thank the organizers today for giving me the opportunity to talk about my work on Parkinson's-linked LRRK2 G2019S mutation. We all know that Parkinson's is typically diagnosed with the emergence of motor symptoms that arise due to loss in dopaminergic neurons in the substantia nigra pars compacta. But what I'm more interested in are these non-motor symptoms that can occur decades prior to diagnosis, suggesting that early pathological changes are occurring within the brain when the dopaminergic system is still relatively intact, implicating other neurotransmitter systems. We're specifically interested in Parkinson's that arises due to mutation in the LRRK2 gene, the G2019S mutation specifically, as it increases kinase activity by 2 to 3-fold. An increase in kinase function has also been observed in idiopathic PD, suggesting that it's a common pathogenic driver. To study this in the lab, we use a G2019S knock-in mouse model, where the mutant protein is expressed in similar levels as that of control, and hence circumventing the deleterious effects associated with overexpressor models. When we look at LRRK2 expression using an in situ hybridization image, we find that it's highly enriched in the striatum and cortex, showing sparser expression in sort of the dopaminergic-rich midbrain areas. In fact, work from our lab shows an increase in spine diameter and an increase in excitatory spontaneous current amplitude in spiny projection neurons, which are the principal neuronal subtype within the striatum, implying a postsynaptic effect. And so the question that I'm asking is, how is this mutation impacting AMPA receptor dynamics at these excitatory glutamatergic corticostriatal synapses? To address this, I first looked at surface GLUA1 and GLUA2, which are the two predominant subunits of AMPA receptors, and found surface GLUA1 was significantly increased in the mutant compared to controlled striatum, with no change in GLUA2, suggesting that this mutation might be impacting AMPA receptor subunit composition. To determine if this had a functional impact, we did whole cell patch recordings, either in the absence or presence of NASPERM, which specifically blocks these calcium-permeable GLUA1 containing AMPA receptors. We found a heightened sensitivity within the mutant T1 synapses, suggesting abnormal accumulation of calcium-permeable or GLUA1 containing AMPA receptors at baseline, which corroborates our biochemical findings. Next, we use super resolution microscopy to look at the distribution of these GluA1 nanoclusters across the spiny projection neuron population and found increases in both synaptic and extrasynaptic areas for the GluA1 nanoclusters. Now, we know surface levels of any receptor are maintained through the antagonistic function of sort of two trafficking arms. So first, looking at endocytosis, where we can differentially tag the surface and internal population, we found increased internalization in wild types at the end of 60 minutes for GluA1 containing AMPA receptors with no change in the mutant, suggesting that there is a defect when it comes to endocytosis. Next, we looked at insertion as well as surface diffusion, which is important when it comes to the synapse. And we did this using fluorescence recovery after photobleaching. And while the wild types are able to recover normally, showing a biphasic association that is both insertion and diffusion for repopulating the bleach spot, the mutant neurons fail to show this. And so what we found was this mutation selectively impairs trafficking of GluA1 containing AMPA receptors. Now, so far I've shown you constitutive trafficking, but what about regulated trafficking? And so to address this, we use a ChemLTP protocol or ChemLTP with AP5, which blocks NMDA receptors, looking at GluA1 surface levels at 5 and 30 minutes. In the wild types, we found a significant increase at 5 minutes that normalizes at 30 minutes. And this is what's been observed in hippocampal neurons as well. In the mutant neurons, we failed to see an increase in GluA1 containing AMPA receptors across the board, with all the 3 treatment groups in the 2 time points, suggesting that there is a defect when it comes to trafficking. So to summarize and conclude what I've shown you so far, at baseline in the wild type neurons, we have both GluA1 and GluA2 containing AMPA receptors, and these levels are maintained through these sort of endocytic and exocytic trafficking arms. With long-term potentiation, there's this drive to insert GluA1 containing AMPA receptors early on at 5 minutes, and this results in sort of normal LTP and healthy cognition. In the case of the mutant neurons, we find that there's this abnormal increase in GluA1 containing AMPA receptors at baseline, driven in part by this trafficking defect, which persists through long-term potentiation, resulting in LTP deficits, which we've observed at corticostriatal synapses in vivo, resulting in cognitive deficits, which we've observed in these mice, which are similar to what has been observed in the Parkinson's patient population. And so here I've provided a cellular mechanism that can help explain some of these deficits that we see early on when the dopaminergic system is still intact. And with that, I'd like to thank Deanna and George, members of the Benson and Huntley Lab, the microscopy core, and our funding sources. I'd be happy to take any questions that you may have. Thank you.
Hi, everyone. My name is Lydia Gramai, and today I'm excited to share with you my recent work on the role of stress response signaling as a systemic regulator of reproduction. So for the last few years, I've been working to understand how an organism systemically coordinates reproduction, which includes multiple events like gametogenesis in the ovary, ovulation and mating directed by the brain, and feeding to finance the energetic cost of reproduction. And all of this must presumably be coordinated by relay mechanisms that can sense nutrient status in metabolic tissues and direct the multiple tissues that are required to coordinate these events. And my work has focused on the signaling events between fat tissue and the ovary, since we know that in clinical settings, abnormal lipid content contributes to infertility. And today I'm going to share with you one mechanism that we found that helps organisms sense metabolic changes within adipose tissue and relay that to instruct reproductive processes. So cells are equipped with a stress sensing mechanism called the integrated stress response pathway, which integrates internal and external stressors to deploy a context-specific response. And this is mediated by activation of stress sensing kinases that induce expression of a transcription factor called ATF4. And likely due to the high metabolic burden of cells like adipocytes, we see a basal level of ATF4 induction under physiological conditions, which we can see here in fixed adipose tissue. So in this work, I used Drosophila or fruit flies to ask and answer the question, what happens to the ovary when ATF4 is lost from the fat? And today I'll share with you a few roles we've identified. The first is that ATF4 promotes the production of yolk in adipocytes that later get trafficked to maturing eggs in a process called vitellogenesis. Now, this yolk is synthesized as lipoproteins, and by staining neutral lipids in fat tissue, we saw a disruption of lipid droplet breakdown in the fat in the absence of ATF4, which suggests that here these lipoprotein carriers for yolk are deficient. And this correlated with less yolk protein abundance in adipocytes in the absence of ATF4, which I've shown here using a GFP fusion protein. Now, since adipose tissue is a major source of yolk for maturing eggs, this also resulted in a loss of yolk granules within the oocytes. And this yolk is a critical source of energy for early embryos, so the loss of ATF4 results in less energy store and likely compromised health of future embryos. Now, in addition to promoting oogenesis through yolk synthesis, I also found that ATF4 in adipose tissue promotes ovulation through the production of neuropeptides. So we discovered that loss of ATF4 causes a block in ovulation, and so we performed a targeted genetic screen of neuropeptides in Drosophila. And we found one called CNMA that is less abundant in the fat tissue of ATF4 mutant females. And we can quantify a block in ovulation by counting the number of mature eggs that are retained within one ovary. And there is a significant increase in the number of eggs per ovary when I deplete CNMA from fat tissue. And I also identified a subset of ovulation-promoting neurons in the brain that respond to CNMA, and blocking the CNMA signal in these neurons was sufficient to cause egg retention as well. And finally, I found that while depleting ATF4 in the fat caused an increase in oocytes retained, simultaneous expression of CNMA in the fat blocked this egg retention, demonstrating that CNMA is at least one of the ovulation-promoting signals downstream of ATF4. And in data that I don't have time to show here, we found that ATF4 plays a nutrient sensing role to alter the rate of reproduction during stress. So amino acid deprivation causes the rate of oogenesis to drop, and then refeeding allows it to rapidly recover. And we found that this recovery following starvation was hindered by the absence of ATF4, suggesting that nutrient sensing by ATF4 is required during this recovery period. So to summarize, I found that ATF4 plays a nutrient sensing role in fat tissue to regulate reproduction on at least two levels. First, by directly promoting oogenesis through the synthesis of yolk, and second, by regulating neuropeptide expression to promote ovulation. And with that, I would like to thank my co-advisors, Deepika and Kauf, as well as the other scientists that helped bring this story to life. And I'm grateful to a number of groups for funding and career development, including the Intersection Science Fellows community for this opportunity. Thank you.
Hi, everyone. My name is Yu Chuan, and I'm a postdoc in the lab with OK from Harvard Medical School. Thank you so much for the opportunity for me to share my work today. I'm interested in using stem cells to build human embryo models and using these in vitro models to study how cells make patterns during development. In my postdoc, I was focusing on somite formation. Somites are epicellulized blocks flanking our neural tube in the early embryo. They give rise to our vertebrae, skeletal muscles, and so on, and they establish the segmental features our body axis. In humans, somite formation occurs extremely early, between 3 to 4 weeks after fertilization. It's a time window when most women haven't realized that they're pregnant yet. So we rarely have access to human embryos at this stage. To overcome this, I turned to human pluripotent stem cells. I use stem cells to build models recapitulating somite formation during human development. One of the models called segmentoid, it recapitulates somite formation in time and space. Somites are formed rhythmically and sequentially from head to tail. Its rhythmicity are controlled by an oscillator called the segmentation clock, with its core gene Has7. Now in this video, you can see that Has7 are oscillating at the posterior end, while the segmental gene MASP2 progresses accordingly. Now, we further use this system to study how the somite patterning. Each somite is divided into anterior and posterior half. By using live imaging, we found that cell sorting analyzes this patterning process. we found that the gene MASP2 is first inducted in a salt and pepper fashion in a forming segment, and then MASP2 high cells are sorted to the anterior compartment, while MASP2 low cells are sorted to the posterior compartment. This is then eventually establishes the anterior-posterior polarity patterning. Now, with this, I'd like to thank my lab and collaborators, and thank you so much for your attention.
Okay, so thank you to the organizers for the opportunity. Today, I'm excited to tell you about a recently published story where we decode and then recode stem cell signaling in the context of lung regeneration and fibrosis. So over the past couple of decades, people have discovered many stem cells, perhaps hundreds across the animal kingdom. And to me, one of the biggest surprises that many, or maybe most of these stem cells are essentially controlled by a small set of very powerful developmental signaling pathways. And so among the most prominent of these pathways is Wnt, which binds to its cognate receptor Frizzle to activate signaling and then controls stem cells in different animals, in different organs within the same animal, and sometimes different stem cells within the same organ at the same time. And so being a powerful pathway, it's often dysregulated or hijacked in disease and has become a major therapeutic target. However, modulating Wnt through broad activation or inhibition turned out to be very toxic because the pathway is active so broadly. And so this then led me to Okay, so thank you to the organizers for the opportunity. Today, I'm excited to tell you about a recently published story where we decode and then recode stem cell signaling in the context of lung regeneration and fibrosis. So over the past couple of decades, people have discovered many stem cells, perhaps hundreds across the animal kingdom. And to me, one of the biggest surprises that many, or maybe most of these stem cells are essentially controlled by a small set of very powerful developmental signaling pathways. And so among the most prominent of these pathways is Wnt, which binds to its cognate receptor Frizzle to activate signaling and then controls stem cells in different animals, in different organs within the same animal, and sometimes different stem cells within the same organ at the same time. And so being a powerful pathway, it's often dysregulated or hijacked in disease and has become a major therapeutic target. However, modulating Wnt through broad activation or inhibition turned out to be very toxic because the pathway is active so broadly. And so this then led me to hypothesize that there must be cell type specific Wnt signaling mechanisms that enable the simultaneous control of so many populations by one pathway. And perhaps by discovering these mechanisms, the basic biology would inform the therapeutic efforts so that we can precisely and safely modulate this pathway for the benefit of patients. And so today I'll tell you about one such effort in alveoli of the lung where gas exchange takes place. And so here, Wnt is known to act on two very different cell types. It can act on epithelial stem cells known as AT2 cells. And in doing so, it promotes their proliferation and in certain contexts, the repair of the tissue. In the same context, Wnt is also known to act on fibroblasts to promote their pathological expansion and the scarring of the tissue, which eventually leads to fibrosis and organ failure. And so in these degenerative diseases that I described to you, such as idiopathic pulmonary fibrosis, people have proposed either activating or inhibiting the pathway based on which cell type they're studying. So they can either activate regeneration or inhibit in the fibroblasts to inhibit fibrosis. So how to reconcile these efforts? Well, what I found is that different cell types in the lung, which all utilize Wnt, actually express distinct receptors for the pathway. And as you can see from this in situ hybridization, the stem cells, which are shown here in red, actually express Frizzled 5 selectively, while the fibroblasts, which are shown here in red, don't express Frizzled 5, but selectively express Frizzled 1. We went on to show through a series of organoid experiments that epithelial cells selectively depend or uniquely require Frizzled 5 for their Wnt-driven stem cell activity, and that fibroblasts use other Frizzled receptors to transduce Wnt. So based on this discovery, then, we would like to activate Frizzled-5 to promote regeneration while circumventing the unwanted effect of Wnt on fibroblasts. And so this is a fine idea in theory, but in practice, Wnt ligands are terrible as pharmacological agents because their specificity is poorly understood and their solubility is simply abysmal. However, people have generated antibody-based Wnt mimetics that allow us to selectively and potently activate any of the 10 frizzled receptors with a high level of potency. And we use these to show that indeed Frizzled-5 is a molecular handle that allows us to selectively engage and activate the epithelial stem cells. And then in preclinical models of lung disease, so here we're showing you bleomycin, we can use these agonists to selectively activate and promote the proliferation of stem cells by about 500 to 600%. This increases the survival of animals in this model by about 50%, and all in while
Hey there, my name is Joseph Swift. I'm a postdoc in the ECRA lab at the Salk Institute, and today I'm going to talk to you about adaptive plasticity in plants. So what do I mean by plasticity? Well, consider human development. It's not very plastic. We all mature and age at roughly the same rate. You could say development is a pretty insulated program that pushes biology along. Not so in plants. So consider Arabidopsis, our model organism. You can see that after 10 days of growth, plant leaves have grown and expanded, but under drought stress, you can see that they remain much smaller. They've restricted their growth. Their growth rate is much slower. In other words, because these plants are experiencing stress, environmental signals are telling the plant, whoa, don't get so big because we don't have the resources we need. This response is adaptive and it acts upon a plastic trait growth rate. So a hypothesis I had was, how do plants signal plasticity in a dose-responsive way? It's not a binary trait, right? So here I'm showing you plants grown under different doses of drought stress, and I'm following shoot area over time. And you can see the rate of growth is quantitative, ranging from fast to slow growth rates. I wanted to understand, what are the molecular mechanisms that are driving these plastic traits? To do that, I took a single nuclei sequencing approach, sequencing the individual nuclei transcriptomes of a bunch of different individual plants across this experiment. So what did I learn? Well, the first thing was that I was able to identify the different cell types of the leaf. Suffice to say that I was able to identify epidermal, mesophyll, and vasculature lineages. And for the sake of time, I'll focus on the epidermis. The epidermis was the most interesting because we were able to see that rates of gene expression within the epidermis were dose responsive with the level of stress, where we found nearly 1,000 genes who had a high rate of gene expression over development under well-watered conditions, but that those rates of gene expression declined as the stress intensified. And indeed, the epidermis cell type correlated significantly with the rate of shoot growth at the physiological level. In other words, what this data suggests is that the cell type, or the skin of the leaf, the epidermis, is actually metering the rate at which plants grow in response to stress. Okay, so I wanted to look at this phenomena in another way, and I wanted to understand if it could help explain how leaf development works under stress. And what's really nice about Arabidopsis is that you have an intrinsic developmental gradient within the rosette. So if you tease apart each of these leaves, you can actually isolate young, temporally young and developmentally young leaves from their much older counterparts. And indeed, there's a nice developmental trajectory that's very loosely analogous to human development between younger and older individuals. And so when I sequenced the nuclei of individual leaves, what I found was that the epidermis, again, naively arranged itself into a developmental trajectory spanning younger nuclei to older nuclei. We can also look at this trajectory in a principal component. So taking genes that are expressed under well-watered conditions, you can see that the principal component of this cassette of genes is dynamic between younger and older leaf cell types. But what was very interesting to see was that as drought intensified, again, we saw a dose-responsive effect where these genes were no longer dynamically expressed between younger and older leaves. And indeed, young leaves appeared more transcriptionally similar to older leaves at the most extreme drought stress. So what does that mean? What that means is that as plants experience drought, it appears that what occurs is that the kinetics of gene expression increase and that it speeds up maturation and causing plants to develop and age more quickly. And so this has implications not only for breeding or engineering plants that are more drought tolerant, but also understanding basic biology of development and plasticity, how environmental signaling may result in creating different phenotypes. And so with that, I'll say thanks for your attention to my PI, Joe Ecker, and my funding sources from Life Sciences, American Australian Association, and Howard Hughes. And I'll take any questions at the end of the session.
Hello everyone, my name is Coral and I'm a postdoc in Rebecca Heald's lab at Berkeley. So today I'm going to give you a little snapshot of how we can use frogs to dissect the molecular mechanisms of genome size control. So if we look across the tree of life, we notice that genome size actually differs really wildly. So between the smallest bacterial genome over here and the largest plant genome over here, there's about a 6 order of magnitude difference. And yet all of these genomes perform the same functions: replication, transcription, DNA repair, segregation of chromosomes. So we really don't understand how genome size influences things like genome organization as well as genome function. And so we can ask this question by comparing across species, but there's actually a simpler way to ask this question, and that's using a single species with a single genome. And we can ask, instead of changes in genome content, we can ask how the physical space being occupied by that genome changes over time. And we know that a time when this is happening is during early embryogenesis. So upon fertilization of an egg by sperm, the embryo will undergo these rapid and successive cell division events, resulting in smaller and smaller cells, as shown here. And we also refer to this stage of development as the blastula stages. I'm showing you this video for a frog embryo, but actually it's really well conserved among all metazoans. So in the past, people have used immunofluorescence to look inside of these embryos. What they found is that structures like the mitotic spindle, as well as the nucleus itself, actually decrease in size as well, scaling with cell size. And my lab in particular, as well as others, have really figured out the molecular mechanisms as well as the general principles that set these changes in size. But what we really didn't understand is the behavior of these mitotic chromosomes, which of course sit at the center of these mitotic spindles, ready to be segregated into daughter cells. And so in my work, I was actually able to show that mitotic chromosomes indeed also scale during this time. And this is primarily through a 2-fold decrease in chromosome length, as shown here. But I really wanted to figure out the molecular mechanism behind this behavior, which is actually for many reasons really hard to do in embryos. So instead we turned to the extract system. So these female frogs can actually lay thousands of eggs overnight. So we can actually just go ahead and collect those eggs in a test tube and centrifuge them to fractionate away the cytoplasmic layer, which is this golden layer in the middle, away from things like lipids and yolk. And the cytoplasm we actually call extract, and really contains the full complement of every molecule and structure that you would find normally in the cytoplasm. But at the same time, it's an extract, it's a biochemical mixture. So we can treat it with all of these different things listed here that in ways that we could really never do in vivo, or at least it would be really, really hard. And the other really special thing about extract is that the maternal genome is actually pelleted away during the centrifugation step. So we can actually go back and add in our own source of genome. So what I've done is actually added back sources of genome that represent either early or late blastula stages of development and went ahead and looked at the mitotic chromosomes. And what I found was that I could actually recapitulate this twofold difference in chromosome length that I had observed in vivo. And so we really think that the egg extracts are able to recapitulate mitotic chromosome scaling, but in a test tube. And so I went ahead and used this system in combination with a bunch of different techniques, as I've shown here, to really dissect the mechanism of this scaling. And so I'm just going to go ahead and summarize this data into a model. So we think that there are actually two developmental cues that scale mitotic chromosomes. One is cell size, and the other is nuclear cytoplasmic ratio. And combined, these cues actually decrease chromosome size by about twofold. And we also know the molecular mechanism involved. And so we think these short chromosomes actually have less condensin 1, which is this DNA looping enzyme. And as a result, these short chromosomes also have bigger DNA loops, which is how we think it can accommodate the same amount of DNA on a shorter axis. And then at the nucleosome level, these short chromosomes also contain increased linker histone H1, which could actually cause increased internucleosomal compaction. So now I've shown you really how chromosomes are scaling in the embryo, but I really haven't talked about why. And that's really a future direction for me. So in general, I'm interested in this question of how do genomes actually adapt to changes in size? And so I've shown you how we can really ask this question in an embryogenesis context using this Xenopus laevis frog. But we can also ask this question on an evolutionary time scale by comparing different species of frogs that span a 6-fold range in genome size. And so at the end of the day, we really want to know what are the fundamental unifying principles behind scaling and really what are the exceptions. So with that, I'll go ahead and thank my collaborators in the Decker lab, my lab, including my wonderful advisor, Rebecca Heald, these two wonderful students who worked with me, and I'll take any questions.
Okay, wonderful. Well, thank you to all the associates for your wonderful talks. And if anybody has any questions, please feel free to paste them into the chat now. We can direct those questions to any of our associates. And while we wait for questions, I am posting a link in the group chat, and you're welcome to vote for your favorite talk. You can vote more than once, and we'll be awarding prizes to the associates with the most votes at the end of the symposium. You can also use this QR code that's now on your screen. We have about 30 minutes for questions. Okay, a question from Coral, and it is for Xiaoteng. How customizable is your platform for non-neuronal tissue?
Thanks for the question. I haven't actually worked on other tissue, but I think about my future plan is to adopt my platform that I developed for other tissues and other cell biologists as well. I think it's pretty adaptive, this platform. And I also want to include in my future work to streamline the generation of the immunoprobes described here. So I think that will really make this is a very helpful tools for other tissues and other cell biology research.
Coral says thanks. We have about 20 more minutes left in this session. I know I just said 30, but I meant 20. And we can take any questions. Anybody has any questions, go ahead and post them into the chat. Okay, we have another one for Xiaotang from Kevin. How many protein copies are necessary for identification in your EM approach?
Thanks for the question. How many protein copies? I actually haven't done any like quantitative measurement. And for my correlated light and electron microscopy work, currently I only focus on the cytosolic proteins because that's easier for the correlation between two image modules. And those proteins, I think, have many copies. That is really easy to identify in both modules. But I want to try in the future whether this approach could also be labeled to be used for label more sparse expressed proteins, like proteins in the axonal terminals, or label some very refined structures of the cells, but that is an open question. Thank you for the question. Oh, sorry, I forgot to turn on my video.
Thank you for that question, Kevin. Okay, we have another question for Krista. In engineering the phage to remove immunodominant motifs, do you expect an immune response to the new epitopes created by smoothing the proteins? Thanks. This is from Ashley.
Well, thanks, Ashley. That's a really good question. I don't have an answer yet, but we are doing a mouse study right now to try to interrogate that. I suspect that we will see more immune responses to new epitopes. I guess we'll figure out if that's a problem down the road, or if those are, less immunogenic and the population of antibodies is reduced overall or not. But it's an open question.
Wonderful question. Thank you. Okay, we have from my co-moderator Deba, for Joseph. Wonderful talk. How do you think aging and stress is correlated in your plant model? Is it comparable with animals?
Thanks for the question. To answer the second part first, I don't know if it's comparable with animals. I would like to, if someone's an expert, that would be really interesting to hear their thoughts. But in plants, I do think that as plants experience stress, they will maybe not correlate with aging, but maybe speed up their development. So plants have very well-defined development programs, from proliferation to expansion to senescence. And senescence is similar to what we see in human aging. We see those signals become more prevalent earlier on as plants experience stress, or at least drought. So there is that correlation there.
Wonderful, thank you. Okay, we have another one from Kevin for Dr. Grimay. Do you think you can, or do you plan to extrapolate to humans from the fly?
Okay. Yes, thank you for the question. We definitely there are definitely a lot of sort of points of connection or conservation between the sort of stress response pathways that we're modeling in flies versus how they sort of work in mammalian systems and even human systems. So in terms of our finding that stress response signaling promotes yolk synthesis. So adipocytes are actually known in mice and humans to be a major source of yolk lipoproteins for maturing oocytes. So that sort of point of interorgan communication between adipose tissue and the ovary is actually a sort of known line of communication. So how ATF4 and stress response signaling impacts yolk synthesis and promotes reproduction in humans is definitely worth sort of future exploration, and sort of broadly looking at how stress response signaling kind of supports reproductive events via adipose tissue signaling, and sort of one of the things that I'm kind of trying to model is how does the adipose tissue kind of communicate with all of these different organs. And so a lot of the obviously tissues and processes we're studying, but even some of the molecular links that we have found so far show gene conservation all the way to humans. So we do think that we're sort of modeling human physiology in multiple ways, and that's something that I hope to sort of continue to expand, both understanding how it operates in homeostasis, but then also how these stress response factors are dysregulated during acute stress or chronic stress, as in metabolic diseases. So these are all sort of future points of study.
Wonderful. Thank you so much, Lydia. Okay, we have a follow-up from Ashley, and I am assuming the follow-up is for Krista. Can you customize treatment by alternating the phages used to avoid immune neutralization?
Good question. This is another one that we don't quite know the answer to. So the mouse studies for phages are sort of just kicking off now, and most of the data that we have has actually been generated in humans that are receiving compassionate use phage therapy. And because of that context, we can't really tweak their treatment in a way that might be like experimental. So we haven't been able to do the experiment that you're suggesting yet, but it's something that we think probably can work. So if you have a cocktail of 3 phages, just give the first one for a month, and then the second the next month, and so on. There are other things that possibly can be more effective in reducing the neutralizing immune response. For example, some preliminary unpublished data so far shows that administering the phage through nebulization and inhalation rather than IV dosing is really effective at controlling like lung infections, but also abrogates the immune response. We can also need to think about things like how the immune response correlates with the burst of phage out of the cells during the course of therapy, and how that might impact things. So there's lots of questions to think about and experiments to do.
Wonderful. Thank you so much, Krista. Thank you for the question. And we have another follow-up for Lydia from Kevin. Why not simply work in mice? Why the fly model?
Yes, thank you. Sorry, in the interest of time, I omitted my sort of why flies pitch from the talk. So I thank you again for the opportunity to sort of advocate for fly research. So I think, your point is well, your question is well taken, that, there's sort of mammalian physiology that might be easier to model or more thought of as more appropriate to model. And obviously there are processes, human reproductive processes, that don't occur in flies. But of course there are a lot that do. And there are a lot that we think that don't, but actually do. So for example, ovulation, which is one of the reproductive events that we found regulated by ATF4. So it might appear that flies don't ovulate the way that humans do, but they undergo a process in flies, which is egg laying, that is at the molecular level and the tissue level very analogous. And in fact, like some of the same sort of neurotransmitters that are known to regulate human ovulation, their analogous neurotransmitter regulates egg laying in flies. And so we have the benefit of sort of having these analogous processes that we can model, but we also have the added benefit in the Drosophila system that there's ample genetic tools for us to sort of understand what's the gene regulation that's underlying these events. We can do genetic screening very quickly. We can make transgenics and mutants in a matter of weeks versus a matter of months. And obviously from like an animal model research perspective, there's just kind of less hoops to sort of jump through to answer the questions. And so I think we can get at a lot of sort of underlying principles in ways that are translatable in terms of mouse and human physiology. We can sort of just answer the questions faster. And I think the repertoire of questions we can answer is a little bit more broad as well.
Wonderful. Thank you so much. And thank you for the question. Okay, next question we have from Coral for you, Xuan. Could you comment on the evolutionary control of segmentation species with different sized spines? Question mark.
Thank you so much for the question. Yeah, like all the vertebrate species, we have like different number of somites, eventually came out to different like characteristics of our spines. So I think this like there are a lot of hypotheses about it. But I think with these in vitro model, especially in vitro models from different species, we can really start to investigate the detailed mechanisms, how these different sized somi, different size somi number are controlled according, like among all the different species. So these are really exciting future direction for the field to go on to answer. Thank you.
Okay. Okay, so while we wait for, oh, here we go. Another question from Deba. Question for Lydia. Since insulin signaling plays an important role in oogenesis and Drosophila, can you comment on how might insulin signaling is modulated in your ATG mutant?
Yes, thank you. That's a really good question. I think there's sort of a lot of a huge body of research that explores the role of insulin signaling, excuse me, both in oogenesis, but also in so insulin signaling is upstream of mTOR, which is known to be very responsive to different stressors. And so mTOR and ATF4, which is part of the integrated stress response pathway, sort of both feed into oogenesis, both during homeostasis and in stress. We actually have recently done some sort of genetic interaction studies to see if ATF4 is employing mTOR in any way in either in homeostasis and in stress, and it seems that they might be acting in parallel. It is known that there's some crosstalk between the two, but that doesn't seem to be at play in the homeostatic role that we've identified. We are still sort of, one kind of outstanding genetic study, which sort of gets more closely at your question, is whether ATF4 is downstream of insulin signaling in any way. So does a reverse relationship hold? And so that's still an outstanding question. But it does seem that both pathways are sort of feeding into oogenesis regulation from adipose signaling.
Thank you so much for the question and for that wonderful answer. So while we wait for more questions to come in, we still have about 8 minutes left for more questions. You can, of course, use the QR code or the link that I posted into the chat to vote for your favorite Associates Flash Talks. And let's see. We also, if we have a pause for questions, we can, Nicole had a great idea of maybe we could allow each of the associates to unmute and say whether or not they're going on the market this year, so that everybody knows kind of where they're headed. And so if any of the associates would like to do that, you are welcome to do that now.
Hi, I'd love to initiate that. So my name's Nicole Perry-Hauser. I am a fourth year postdoctoral fellow, and I am tentatively tipping my toe into the market this year. I'm trying to do a more targeted approach as I await the decision on my K99 application, which I'm sure many people here can agree is a very stressful period of time. But yeah, it's been a great opportunity to be here. I think the symposium is an excellent way to meet different people in the community and to connect.
Thank you so much, Nicole. If you'd like to speak a little bit more, just please turn on your cameras if you're gonna let us know.
Hi. Oh, sorry. Yeah, thank you for the platform. And my name is Chuan, and I'm also I will be on the job market this year, and I'm interested in continuing using stem cell to build and learn how our human body developed. And thank you again for your attention and questions.
Yeah,
Sorry, no, go ahead.
Okay, thanks, Lydia. Yeah, I'm just in a similar situation as what Nicole described. I'm a fourth year postdoc at Icahn School of Medicine at Mount Sinai, and I'm starting to dip my toes in the job search as well, again, taking a targeted approach. And I will be focusing on more homeostatic plasticity and its role in neurodegenerative diseases and aging. Thank you.
Thank you.
And yes, I'm Lydia. So I'm a fifth year postdoc at the University of Pittsburgh. I'm also jointly advised by Kaf Jaraza at Duke University, but I'm housed in Pittsburgh, and I am on the job market this season. So as you know, some of my Q&A may have alluded to, I'm using Drosophila as a model to investigate the interorgan regulation of reproductive events. And that's, you know, the sort of first branch of that is going to be via stress response signaling and this transcription factor ATF4, how it regulates reproductive behaviors like ovulation and feeding, as well as how steroid hormones might be regulated downstream in order to regulate oogenesis and other related events.
Wonderful. Thank you. Thank you. Would any of our other associates like to unmic and say what they're up to this year?
I could share a little bit. My name's Krista. I hope that my text is no longer reversed. Kelsey told me it was backwards.
It's perfect now.
I got it now. Yes, I'm also on the job market this year. I'm hoping to find a position, preferably, well, we'll see, in a biophysics department, continuing to research phages and to use the structural analysis of phages to help us learn to make better ones that are less immunogenic or have broader host range, and to learn how they work better.
Thank you. Thank you, Krista.
Maybe I'll go. So my name is Ahmad. My photo in my talk was very small, so maybe you didn't see what I look like. But I am on the job market this year, and my hopefully future lab will work on following up on some of the Wnt mechanistic dissections I talked about, but also will be developing chimeric organoid screens to essentially decode how stem cells and their niche communicate.
Awesome. Thank you.
I guess I'll follow up on Ahmad. I'm Jared. I'm also going on the market this year, a little more targeted for family reasons, but hoping to start a lab kind of around the idea of using multi-omics to explore new targets for antimicrobial resistance. And then also because my postdoctoral lab works in the gut microbiome and my background's actually in lung biology, hoping to work more in the pulmonary microbiome as well.
Wonderful. Thank you. Thank you. We have a couple more minutes. Any other associates or any other questions before we take our break? Okay, well then I will take this opportunity once again to say thank you to all of our associates for your wonderful talks. Thank you to everybody for your attention and your wonderful questions. And we will take a break now until about 2:15, and then we'll have the next session, which is panel 1, strategies to ace the job market. Thank you all again for coming. Have a great one.